The overall goal of this procedure is to assess cognitive and emotional functions of interacting brain structures through simultaneous recordings from both deep and surface neuronal sources.
This method can help answer key questions in human neurophysiology, such as the relationship between brain activity patterns and brain function, especially regarding deep-seated, barely accessible brain areas.
The main advantage of this technique is that it uses the framework of deep brain stimulated patients to extract live information of cortical and subcortical structures in humans during cognitive and emotional tasks.
The recording steps in this section focus on a patient with a movement disorder performing a flanker task. The analysis steps in the next section, however, focus on a patient with chronic disorder of consciousness listening to a cognitive emotional speech paradigm.
Begin by setting up the electroencephalography or EEG equipment in the testing room. Connect the recording computer to the EEG system and start the EEG recording software. Click File and then New Workspace to define the workspace in the EEG recording software by specifying a sampling frequency of 5 kHz, a low cutoff and high cutoff frequency, EEG channels according to the International 1020 system, and local field potential or LFP channels. Click Monitor to verify that the specified channels are now